DNA replication is the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having three phases: initiation, elongation, and termination. Let's begin by considering the initiation phase. For copying to take place, the double helical structure of the DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, a DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble. This establishes two replication forks that will move away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis. Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand. A DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome. The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA and encircle it, the clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring. The opened ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase. The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end. During elongation, the replication bubble gets bigger as the helicases move apart, establishing two replication forks that move in opposite directions. The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose single strands. Let's focus on just the right-hand fork for the moment, as the processes at both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the two strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. It's always made in the 5' to 3' direction. Because the replication of the two DNA strands is coupled, the leading strand is copied continuously, while the lagging strand is copied in short stretches called Okazaki fragments. Each Okazaki fragment is initiated by a primase that synthesizes a short RNA primer. This primer is later elongated by DNA polymerase. The single-stranded DNA between the newly made Okazaki fragments is bound by single-stranded binding protein, or SSB. In order for the two polymerases to move together with the helicase as the fork moves, the DNA template for the lagging strand must be looped around so that both polymerases are moving in the same direction. With the polymerases now coupled, DNA replication can proceed coordinately on both the leading and the lagging strand. Once the lagging strand polymerase reaches the RNA primer from the upstream Okazaki fragment, the lagging strand loop is released, and Okazaki fragment maturation and ligation by polymerase I and DNA ligase occurs. While this maturation is occurring, the primase enzyme that is also travelling with the fork as part of the replisome initiates a new RNA primer on the lagging strand. Once this primer is made, the lagging strand polymerase on the replisome, having completed synthesis of the previous lagging strand, then re-engages with the 3' end of the new primer, forming a new loop on the lagging strand. DNA synthesis thus proceeds with the leading strand polymerase continuously engaged with the DNA and the lagging strand polymerase engaging and disengaging as each Okazaki fragment is synthesized. To synthesize the lagging strand, the DNA template is looped out while it is coupled to the reprosome complex, and the loop is released after each Okazaki fragment is completed. The length of the looped DNA at its maximal size is therefore the length of an Okazaki fragment, which in bacteria is known about the details of the termination of DNA replication. Once the entire chromosome is copied, the two replication forks meet each other and are dismantled. The ends of the new DNA strands are then joined, again by DNA ligase. The two daughter DNA molecules now consist of one parent strand of DNA shown in grey and one newly made strand shown in green. Thus, DNA replication is termed semi-conservative.